Okay, hi guys. We're just going to pick up exactly where we left off on the neuroanatomy section. We were talking about the different functional areas of the brain. So we'll just continue on with that. Another functional area of the cerebrum is the olfactory cortex or olfactory area. Olfaction is just smell. So the olfactory area is the area that receives olfactory impulses from the nasal mucosa. Um, Originally from the nasal mucosa. These allow us again to have the taste of smell. The olfactory cortex is on the medial side of the temporal lobe. So if you look at the bottom picture, remember the temporal lobe is this lobe right here, the lobe that's underneath this fissure or deep sulcus right here. You have to actually kind of pull back the temporal lobe and look on the inside of the temporal lobe or the medial aspect of the temporal lobe to get to the olfactory cortex. Another structure or another functional area that's also on the medial aspect of the temporal lobe, it's just on the medial and inferior side of the temporal lobe, is the hippocampus. The hippocampus is an important part of the limbic system, which we said was the emotional brain. If you look at this picture here on the top right, the area that's blue right here, these are the hippocampal gyri, or the area that we would refer to as the hippocampus. So again, it's on the medial aspect of the temporal lobe. It's just on the inferior side, whereas the olfactory cortex would be more superior. The hippocampus, again, is an important part of the limbic system, and the limbic system is what we refer to as the emotional brain. It's highly important in our emotions as well as in memory. The limbic system also has some autonomic functions in that it controls the like the actual physical response that we have in the body in response to emotion. That's really via the hypothalamus, but the limbic system in general, which includes the hypothalamus, does control our responses to emotion. So that's why when you are, really worked up emotionally, you'll also have a sympathetic response that occurs, right? Your heart rate, your blood pressure change when you're really worked up emotionally. That's because the limbic system is connected to or associated with the autonomic nervous system. The hippocampus, again, has important functions in memory, specifically short-term memory. Long-term memory is stored in other regions around the cerebrum. And it actually looks as if, depending on the type of memory, it can be stored in different areas of the cerebrum. But that's long-term memory. Short-term memory, which is like being able to take in all of this stuff that's happening right now and convert it into something that you can recall, an hour from now. That's short-term memory. And that relies heavily on the hippocampus. So for example, people who have hippocampal lesions have difficulty forming new memories. However, with a hippocampal lesion, old memories, so memories that are already established and stored in long-term storage, are unaffected. So old memories are not affected with hippocampal lesions, but the ability to form new memories is affected. That's what the movie 50 First Dates is trying to show you. In 50 First Dates, like remember the girl she'll go on a date with Adam Sandler, and then the next day she doesn't remember any of it, and it's like she's meeting him all over again. What they were depicting is, I mean, in kind of a silly, upbeat way, they were depicting somebody with damage to the hippocampus. So next, the visual cortex and the visual association area. The visual cortex and visual association area are back in the occipital lobe, so back in the very, very back of the brain. The visual cortex, or primary visual area, is in the very, very back of the occipital lobe, so in the posterior occipital lobe, and it extends medially in the walls of the calcarine sulcus. So if you look at this bottom picture right here, right, you can see in green here, this is the primary visual cortex. And this is where we receive signals regarding sight. The visual association area is literally just kind of wrapped around and surrounding the visual cortex. So the visual association area will be like around here, and then also it does extend in medially like this. Okay, so it kind of surrounds the primary cortex. The visual association area allows us to understand sight, and it's an area where we store visual memory. Remember, we said long-term storage of memory can be in different areas of the cerebrum depending on, the type of memory that it is. So the storage of visual memories would occur here in the visual association area. And then again, understanding what it is that we're seeing. So the raw data, the just basic data from sight, right, about sight goes into the primary visual cortex. The visual association area is where we can appreciate what it is that we're looking at. So understanding of shapes, color, depth perception, all of that occurs in the association area. So remember that fibers are areas of white matter in the brain, and that this white matter contains axons which carry signals between neurons. Or from the presynaptic neuron to the postsynaptic neuron. The message that's going to be sent to the next neuron is carried down an axon. And we have fibers that are present in the brain. We have these, they're also called tracts. In some cases, like projection fibers are called tracts as well. But these are just areas where we have a lot of axons that are carrying signals from one neuron to another neuron. So these fibers are how different parts of the brain communicate with each other. We classify fibers based on where they're going, like how far away are they actually carrying the signal? Are they carrying the signal just from one gyrus to the neighboring gyrus? Are they traveling between the two hemispheres? Are they traveling down to the brainstem or even further down to the spinal cord? This is how we classify fibers, just based on where they go. Commissural fibers connect similar functional areas between the two cerebral hemispheres. So commissural fibers go from between the left and the right hemisphere. And this just facilitates transhemispherical transfer of information. So this is just allowing for communication between the left and right hemisphere. So the two sides of the cerebrum know what's going on with each other. The largest of the commissural fibers is the corpus callosum. We looked at the corpus callosum in the first part. Remember if you separate the two hemispheres and you look on the medial aspect, there's this like large curved white area right by the lateral ventricle. And that is the corpus callosum. The corpus callosum contains a ton of fibers that are going back and forth between the two hemispheres. Those are commissural fibers. Association fibers connect various parts of the same hemisphere. So they remain in the cerebrum. Same thing with commissural fibers. Commissural fibers remain in the cerebrum. They don't leave the cerebrum. So association fibers remain in the cerebrum and they remain in the same hemisphere. So they're either going, they're either in the left hemisphere or they're in the right hemisphere. They're not crossing. There are two different types of association fibers. There are short association fibers, or they're also called arcuate fibers. But short association fibers are short. They connect areas that are right next to each other. So for example, from one gyrus to a neighboring gyrus, and so just a little tiny fiber. Long association fibers connect distant areas within the same hemisphere. So an example of that is from one lobe in the hemisphere to another lobe. Okay, so from the frontal lobe to the parietal lobe, from the parietal lobe to the occipital lobe of the same hemisphere. Those would be long association fibers. Projection fibers, I don't like the way that this is worded, the beginning of this, but projection fibers connect the cerebral cortex with another area of the brain. Maybe that's a better way to say this, that projection fibers connect the cerebral cortex with another area of the brain. So they can connect the cerebral cortex with areas in the brainstem, with deep nuclei, with the spinal cord, with the cerebellum. They're leaving the cerebrum, essentially. Okay, the other fibers stay in the cerebrum, but projection fibers project down to lower areas of the brain and brainstem and spinal cord. There are tons of projection fibers, or again, we call them tracts, so like motor tracts, which are efferent fibers leaving the brain. There are sensory tracts, which would be afferent fibers, which are entering into the brain. And there are just tons and tons of these examples of of projection fibers. Here you just see examples of all three types of fibers. In this first picture, it's telling you it's a corpus callosum. So right off the bat, that should tell you that these are commissural fibers. And then also you can see they're going from one hemisphere to the other hemisphere, right? They are crossing over between the hemispheres. This next picture, the middle picture is showing you the two different types of association fibers. So the brain has been sectioned, it's been cut in half, and you're just looking at one of the hemispheres, right? So you're looking at the left cerebral hemisphere. And you're just, like, these fibers are just traveling in the cerebrum, right? They're not extending down. You don't see the diencephalon or the brainstem or the spinal cord. You can see like little short fibers, for example, right here. Long fibers would project down between the lobes. So you're going from, in this case, the parietal lobe back to the occipital lobe. Okay. All association fibers because they're remaining in the cerebrum and in one hemisphere. The very bottom picture is showing you projection fibers. Again, this has been sectioned. You can see all of this up top is the cerebrum, but then if you look down, you can see here, this is the diencephalon, where we have the thalamus, hypothalamus, epithalamus, and then continuing down from that would be the brainstem, and then the spinal cord. And you can see these fibers, right, are going from the cerebrum, and then they're going down into, parts of the diencephalon. Okay, parts of the diencephalon, right, they're extending down to the brainstem. So they're leaving the cerebrum. If the fibers or the tracts, if the axons are leaving the cerebrum, then they are projection fibers. They're projecting down to another area. Okay, so here we see examples, again, of all of the different tracts and the different types of association fibers, commissural fibers that are present in the brain. And then we can kind of talk through this and see why these are so important. And really, in short, because nothing we do is simple. The brain is very complex, and our actions, activities, thought processes are very complex. So we need all different areas of the brain and brainstem in order to accomplish things. And the only way for neurons in different areas of the brain and brainstem to communicate with each other is to send messages via their axons, the white matter, right? That's these fibers that we're talking about. If you look at this top picture here with the red, these are showing you association fibers. Okay, so short fibers, for example, up here, right, just connecting little neighboring gyri with each other. You see some of the large association fibers that are present. For example, this is the superior longitudinal fasciculus. And then down here in the bottom, you see the inferior longitudinal fasciculus. The uncinate fasciculus is right here. The cingulum is right here, the cingulum fasciculus by the cingulum gyrus. The corpus callosum is not an association fiber. So here you can see the corpus callosum right here, but that is not an association fiber. That's why it's not in red. Remember, that's a commisurate fiber or commissural fiber. So you can see here in the center, if you look at this picture here on the left, you can see the two cerebral hemispheres. And then in the center of the two hemispheres, you see that they're connected with each other via the corpus callosum. In this bottom picture here, this is showing you the projection fibers. That are bringing messages to and from the cerebrum. In this case, these messages are going to and from the motor cortex, right? You look at the cerebrum. Remember, the precentral gyrus is the primary motor cortex. And you see there's information going to and from that motor cortex. The information going in is shown on these green lines. So those would be afferent fibers. You see the information is coming in from, for example, the cerebellum. This down here is the cerebellum, and the cerebellum is important in controlling or important in allowing us to improve repetitive movements. So like learned movements that you do over and over and they get better and better and better. The cerebellum is important in that process. So you can see the cerebellum is sending signals up to the motor cortex to give it help, right, or give it instructions on how to improve the signals that it sends out the next time. And then in blue, you see the efferent signals, which will eventually be sending, commands out to skeletal muscles to tell them to contract. So these fibers you can see are coming down. There's numerous different relay stations, right? Each, like you see all of these little synapses that can occur. But ultimately, you see that information comes down to these nuclei in the brain, and it makes its way down to the spinal cord. So it's important that these projection fibers are present because something like movement involves all different areas of the brain. So the primary motor cortex is the one that's sending the signals down to tell the muscles to contract, but there are other areas that are monitoring those signals. So other areas also have to get that information. And then again, this primary motor cortex is not perfect. It needs input to help it figure out what to do. So it's important that the cerebellum can send signals up in order to help it adjust future movements. If you look in this picture on the left, it's interesting because you can kind of see like how many different areas would be involved with something as simple as like speaking a word that you read, right? If you have, a book in front of you and you're reading aloud, just reading that word and then speaking it aloud involves areas all over the brain. Also, it's kind of interesting if you look at the visual cortex here, you can see that for each eye, there's a right field of vision. That's what you see in blue. And then in red, you see the left field of vision. And the fibers that are carrying signals from the eye, half of those fibers will crisscross. So the fibers that are coming from the right field of vision, those from the right eye, those are going to cross over to the left. Right? And then the fibers that are carrying the left field of vision, those are just going to continue on down to that right side. If you look at the left eye, on the left eye, the right field of vision, those fibers are not going to cross. They're going to continue. But the fibers that are carrying the left field of vision, those are going to cross over to the right side. So if you look at the very back here at the visual cortex, you see that the right visual field from each eye is on the left hemisphere, right? And the left field of vision from each eye is in the right hemisphere. This is the point where, sorry, my pen just keeps going out on me. I hope it'll last the rest of the semester. The point here in the front of the brain where they cross, where half of those fibers from each eye are going to cross, that's the optic chiasm, right? The optic chiasm that you see on the inferior side of the brain. Okay, but anyways, something as simple as speaking a word that we read, right? First, you have to see the word. So the information goes from the eyes back to the primary visual cortex. But the primary visual cortex is just getting that raw data. Wernicke's area is an important association area for language and reading. So the information is going to have to be transmitted to Wernicke's area for you to be able to actually read that word. And then if you want to speak the word, then Broca's area is going to be involved, or the speech center is going to be involved. So the signals will have to get sent to the speech center so you can figure out how you're actually going to form the word. But then it's the primary motor cortex that actually sends those signals out to the muscle, right? So the Broca's area, or the speech center, is going to have to give that information to the motor cortex. And then the motor area is going to have to send the signal out to the muscles to contract. Okay, so you see something as simple as just, saying a word that you read involves numerous different areas of the brain. Okay, so we'll talk about brain lesions and how they affect the body, right? Like what symptoms you would see with lesions from different areas. And if you know what each area does, then you can kind of guess what a lesion there would do, right? It would interrupt whatever that function was, whether it was movement or planning movements or personality or whatever. So first, we'll talk about some frontal lobe lesions. Remember, the frontal lobe has numerous different functional areas in it. It's got the motor cortex, the premotor area, Broca's area, or the motor speech area, the prefrontal cortex. There's a lot going on in the frontal lobe. So depending on where the lesion is, the symptoms or the difficulties that the patient has can be very, very different. Lesions in the motor cortex interfere with the brain's ability to send voluntary signals out to the skeletal muscles. Hey, remember, the motor cortex is in the precentral gyrus, just in front of the central sulcus. And remember that the motor cortex is where the upper motor neuron cell body is. And again, this is where all voluntary skeletal muscle contractions originate, except for some reflexes. But this is where they originate. Also, keep in mind that the motor neuron is controlling the opposite side of the body, right? So remember that it crosses over. So the left hemisphere is controlling the right side of the body, the right hemisphere is controlling the left side of the body. So damage to the upper motor neuron in the motor cortex results in contralateral symptoms. So symptoms on the opposite side of the body. And those symptoms can range from spasticity, exaggerated tendon reflexes, all the way to complete paralysis. Okay, again, this is where the signal to contract originates. So if there's a lesion that's destroyed those neurons, there is no signal to contract that's going to be sent to that muscle. So it can result in complete paralysis of whatever muscles those upper motor neurons were controlling. Also, keep in mind that the motor cortex, that specific parts of the motor cortex control specific parts of the body. Remember, we looked at the guy who had his legs hanging down into the longitudinal fissure, and then he was laying upside down. And we said that different areas of the cortex control different parts of the body. So you can look at where the lesion is and see, what part of the body you would expect symptoms in, or vice versa, right? If you have symptoms in one part of the body, you can say, okay, I can predict where this lesion is going to be based off of that. If the lesion is in the premotor cortex, this does not result in paralysis, because you still have the upper motor neuron that can send the signal down to the muscles to tell them to contract. So there's not paralysis. The muscles can still contract. It's just that they don't do it in a very good way. They don't contract in a way that necessarily makes sense. So this leads to the inability to execute learned voluntary movements, and that's called apraxia. Okay, so, and again, without paralysis. So the person knows what they want to do. They consciously are aware of what they're trying to do. It's just that they can't quite make their body behave the right way. And the reason for that is, remember, the premotor cortex is the area that stores the plan, right? That's the area that says, okay, this is the blueprint for how we're supposed to contract, or what signals we're supposed to send out every time we want to do, whatever, a cartwheel. So without that ability, without the plan, the motor cortex sends out signals, but they're not mastered, right? It's like the first time you tried to do something. It's really messy and sloppy, and it doesn't work quite right, because again, you've lost your plan, which is in the premotor cortex, but no paralysis. If the lesion is in Broca's area, remember, Broca's area is also called the motor speech area. And this is the area that helps us plan out the contractions that are necessary for speech, right? Really complex tongue movements and movements of the lips. These are planned in Broca's area. So if the lesion is in Broca's area, this results in expressive aphasia. So can't speak properly. but they comprehend perfectly. Like the person has comprehension. They understand what you're saying to them, and they know exactly what they want to say back to you. So there's no deficit in learning or understanding. The deficit is just that they can't make their mouth essentially do what they want it to do. So they just can't speak. They can't say the words. This is also sometimes called apraxia of speech, because apraxia is the inability to execute learned movements of speech. So apraxia of speech is the same thing as expressive aphasia. If the lesion is in the prefrontal cortex, this results in big changes in behavior and personality. Remember, the prefrontal cortex is like how we know how to behave. This is like what makes us human, right? This is your personality, the way that you kind of look at a social situation and judge the way that you should behave. So lesions in the prefrontal cortex can make a person just completely change. Results in unacceptable social behavior, disturbances of judgment, so like inappropriate judgment calls, changes in hygiene. So somebody, isn't showering anymore, they're not brushing their hair, they come in just kind of dirty with rumpled clothes because the area that tells them, hey, that's not acceptable, isn't working. Changes in sexual behavior, so like extreme, somebody all of a sudden being very promiscuous when they weren't before at all. And again, personality changes. So all of this occurring is a sign that there might be a prefrontal cortex lesion or some issue with the prefrontal cortex. Okay, parietal lobe lesions can be associated with prosopagnosia, which is the inability to distinguish faces. This is also seen, though, or probably more seen with temporal lobe lesions. There's a gyrus called the fusiform gyrus, which is utilized for facial recognition, and that goes from the temporal lobe back to the occipital lobe. And lesions in that area are very commonly associated with prosopagnosia. But the parietal lobe is also involved in facial recognition. So lesions here sometimes are associated with prosopagnosia. But again, we'll talk about this again in the temporal lobe coming up next. Remember that at the very beginning of the parietal lobe, in the precentral gyrus, we have the primary sensory area or primary somatosensory area. And this is where we receive signals about touch, right? So touch, pressure, pain, temperature, all of those signals go to the primary sensory area in the precentral gyrus. If there's a lesion there, that results in sensory loss, right? So the loss of the sense of touch, right? Touch, pressure, temp, pain, etc. And this is a contralateral loss. Okay, so if the lesion is on the left side, the loss of sensation is going to be on the right side of the body. If the lesion is in the sensory association cortex, this results in agnosia. There are all different types of agnosia, right? Up here we talked about prosopagnosia. There's, tactile agnosia, which is what we're talking about here. This is tactile agnosia. Agnosia is just the inability to process sensory information. So you still have the awareness of the sensation. So a lesion in the association cortex, you can still feel that something is touching you. You're aware of something, but you don't make sense of it. You don't really understand what it is that's touching you. You lose the significance. So sensation remains, but the significance of it is lost. So if your eyes are open and you can see, you can kind of come up with what's happening. But if your eyes are closed, if you can't see it and you're just feeling something, you don't understand what it is that you're feeling. You can say, yes, something is there, but I don't know what it is or why it's happening. This can be associated if we have sensory lesions in the primary sensory area. This can be associated with something called hemi-neglect. Hemi just means half, right? So this is half neglect. This is neglect of half of the body. This is with like severe lesions in the primary sensory area. The person doesn't sense half of their body. They don't have that feeling of the body there. So they don't do anything to it. They don't pay attention to that half of the body. They don't take care of that half of the body. The one half of the body ends up just being neglected because there's not a sensation that it's there. Lesions in Wernicke's area, which remember is a like an association area for or an integrative area for multiple other areas. Wernicke's area lesions can result in receptive aphasia. Receptive aphasia is when you can't comprehend written or spoken speech, but you can speak. Okay, so if somebody is speaking to you, you don't understand what they're saying to you. If somebody writes something down, you can't read it and understand what it's saying. You can't take anything in, but you can speak. Okay, so you don't lose the ability to speak. You just can't receive that speech. Alexia is the inability to read and understand. Agraphia is the inability to write. So like alexia, inability to read and understand, you can still see. So you can look at a page and you can see that there's stuff on it. You even know that that's writing on a page, but you can't read it. You don't understand. Now you can write. So with alexia, you can't read something, but you can write. Agraphia is the inability to write, okay? And again, these aren't like intellectual deficits. You could do these things previously. You just can't do them now with parietal lobe lesions. If the lesion is in the lower part of the post-central gyrus, remember the post-central gyrus is mostly primary somatosensory cortex, but at the very bottom of it, remember we had the gustatory cortex. And gustation is taste. This is where we receive information from the gustatory receptors or taste receptors on the tongue. So if there is a lesion in the gustatory cortex at the bottom of the post-central gyrus, then that can lead to a loss of the sensation of taste, which is ageusia. A-G-U-E-S-I-A, loss of taste. Okay, so temporal lobe lesions. Temporal lobe lesions can be associated with partial hearing loss and auditory agnosia. Auditory agnosia is when you can't respond appropriately to complex sounds. Okay, so you don't understand the significance of the sounds that you're hearing. The reason that this is partial hearing loss is, remember, you have two ears, you have a temporal lobe on both sides, so you have primary auditory cortex on both sides. So the lesion is only going to be on one side. So you would have hearing loss on that side, but not the other side. So it's not complete hearing loss, just partial hearing loss. The auditory agnosia again, is because of the auditory association area where you understand and make sense of sounds. You lose that ability with a lesion in this area. If there's involvement of Wernicke's area, again, that would result in receptive aphasia. We mentioned that with the parietal lobe, but receptive aphasia, remember, is when you can't understand what's being spoken or you can't understand what's written. Prosopagnosia can occur, again, which is face blindness. And this is specifically if the lesion is in the area of the fusiform gyrus, which is on the inferior aspect of the temporal lobe and occipital lobe. The fusiform gyrus is the area for facial recognition. So lesions in this area make us unable to recognize faces that we are familiar with. Now, you still know the person. You still have memories. You know that they're important to you. You could recognize their voice. But if you were shown a bunch of pictures, you couldn't pick out the person from the pictures. You don't recognize their face. Remember, the hippocampus is on the medial and inferior side of the temporal lobe. So if the hippocampus is involved in the limbic system and really important for memory. So if a hippocampal lesion is present, the person can lose the ability to form short-term memories. So the long-term memories that they have already are there. They're not affected. So there's no retrograde amnesia. They still remember everything from before. But there's anterograde amnesia. They can't form new memories because they have no short-term memory. And with memories, you have to have first short-term memory, and then that gets converted to long-term memory. If you can't take stimuli and convert them into short-term memory, you can't put them into long-term memory later. So there's anterograde amnesia, or an inability to form new memories with hippocampal damage. Involvement of the limbic areas. Remember, again, we said the limbic system was the emotional brain. So if the limbic areas are involved, it results in changes in emotion, as well as changes in autonomic functions. The limbic system, again, is involved in the autonomic nervous system. That's why emotional stress can turn into physical symptoms because of that connection between the limbic system and the autonomic nervous system. Lesions in the occipital lobe primarily affect vision. Remember, the occipital lobe is important, or the occipital lobe has the primary visual cortex in the very back, and then the visual association area surrounding it. Remember that there's the left hemisphere and the right hemisphere, and that each of them is responsible for half of the field of vision, right? So like the left hemisphere receives information from the right field of vision from both eyes. And then the left hemisphere, so the left hemisphere gets info from the right field of vision, and then the right hemisphere gets information from the left field of vision from both eyes. So if it's only one occipital cortex is involved, then you just lose half of the vision, right? You lose the opposite field of vision. Again, you don't lose vision from one eye, you lose half of what that eye sees, right? And half of what the other eye sees. If both, this should say visual cortex. Sorry, it shouldn't say occipital cortex, it should say visual cortex. If both of the primary visual cortex cortices are involved, then there's a complete loss of vision. Because this is where the information from your eyes, from your receptors, goes, right? It goes back to the visual cortex, and that's where you get raw data about sight. So if those areas are damaged, you don't receive that data, so you don't see anything. If the visual association area is what is impacted, then you can see. You still have raw information coming into the primary cortex, but you can't make any sense of it. So that results in visual agnosia, which is an inability, so you can't interpret what is seen. So other things that are lost in this case are the recognition of motion. So because motion is like putting together multiple things that you're seeing and making sense of them, right? Something is here, then it's here, then it's here, then it's here, and you have to put all of those pictures together to understand motion. And you can't do that. The association area does that. It combines the pictures to make sense in equal motion. So you lose the ability to recognize motion. Also lose visual memories. So, you know, if there's a painting that you love, you lose the memory of that, or things that you can picture. There are memories where you can just picture what was happening at that time. Those are visual memories. And remember, long-term memories are stored in all different areas of the cortex. So visual memories, it makes sense, are stored in the visual association area. There's also a loss of color vision because the appreciation of color is in the association area, and then an inability to carry out visually directed movements. You'd be surprised how frequently we sign things, right? We tell people what to do by pointing or whatever. Or, you know, there are street signs that don't have words on them. They're just pictures. And you lose the ability to recognize what that means to you. Or again, visually directed movements. Like if I point at something right now, you know to turn and look at it, but you wouldn't understand what that meant with lesions in the association area of the occipital lobe. Okay, so technically, Alzheimer's is a disease that affects the brain, right? And I'm sure we've all known somebody who has Alzheimer's or seen some pictorial representation of Alzheimer's disease. And I think the thing that we think of first is forgetfulness, right? Or a loss of memory, and then you don't recognize people as you deteriorate. So loss of memory is a major impact of Alzheimer's disease. Technically, Alzheimer's disease occurs because there's an increase in beta amyloid proteins, and it ends up, or plaques, and what we call neurofibrillary tangles are seen in the brain, and this disrupts the normal neurons, right? This disrupts normal neuron function. There's also a decrease in some neurotransmitters, like a decrease in acetylcholine, that is seen because the enzymes that are used to produce the acetylcholine become deficient. So then we see, you know, not enough acetylcholine present, which also interrupts, you know, interneuronal communication in the brain. In general, Alzheimer's is a degenerative disease, and it's associated with brain shrinkage in the hippocampus, as well as several association areas. The hippocampus, you guys know, is important for memory, right? Especially forming short-term memories and forming new memories. So this is why with Alzheimer's disease in the beginning, we have this early forgetfulness that occurs, you know, forgetting where you left something, forgetting where you parked, you know, forgetting where you're supposed to be, where you're supposed to go that day, where you put things. That's because of shrinkage and degeneration of the hippocampus. And again, memory loss that occurs because in order to form memories, you need the hippocampus to be active. There is a gradual increase in the restriction of daily activities that occur as the damage spreads. There's changes in personalities that occur, changes in mood and behavior. And again, this all gets progressively worse as more and more of the brain loses function. Speech can also be affected, especially as you get, you know, more severe in the disease. Here you guys can just see kind of a demonstration of the shrinkage of the brain that happens in Alzheimer's disease. And again, there's really severe shrinking of the hippocampus that occurs, but there's also shrinkage of the cerebral cortex that occurs as well as the disease progresses. And just in general, this picture on the right, you can see a nice healthy brain, and then you see the shrinkage in the second picture. You can see nice, healthy neurons with all of their, interconnections. And then in the picture on the right, you can see that the neurons look terrible. And instead, you've got all of these big tangles and plaques that have built up, and those are interfering with, the ability of the neurons to actually communicate with each other. The picture on the left, first you see a nice healthy brain. You can see, the lateral ventricles. The cortex is full, right? Like there are little fissures and sulci that are present, but the cortex is taking up a lot of space there. In the little smaller picture, you can see the hippocampus, right? Just underneath the very tail end of the lateral ventricle. Right, you can see the hippocampus right here. Remember, it's on the medial and inferior aspect of the temporal lobe. If you look at mild Alzheimer's disease, there has been shrinkage of the hippocampus, which means that the ventricles appear larger, right? So the ventricles are the spaces, and remember, it's the hippocampus is right up underneath the ventricle. So as the hippocampus shrinks, that means there's more open space, there's more ventricle. And you can see that everywhere, right? Here in the center, you can see that as well. And then severe Alzheimer's disease, you can see the ventricle is really, really large and the hippocampus is tiny. Same thing with the cortex, right? All of these cortical regions are just completely shrunk, so that the fissure now looks almost like a ventricle, right? Like this fissure, the longitudinal fissure is huge. Because there's been so much brain shrinkage that occurs as cell death occurs. Okay, so we're going to move on from the cerebrum to the cerebellum. The cerebellum is the little area of the brain that's like the posterior and inferior to the cerebrum, but the cerebellum is located in the posterior cranial fossa below the tentorium cerebelli. The tentorium cerebelli, remember, is a dural fold, right? An infolding of the dura mater that kind of goes into the fissure, the transverse fissure, and then curves back out again. So inferior to the tentorium cerebelli is the cerebellum. It forms the roof of the fourth ventricle. The fourth ventricle is located between the cerebellum in the back and the brainstem in the front. And it's located just dorsal or behind the pons and medulla, which are part of the brainstem. Structure of the cerebellum. The cerebellum is smaller than the cerebrum, but it's kind of similar in that it's folded to increase surface area so that we can pack more neurons in there, and that we've got gray matter on the outside, white matter on the inside. There are two hemispheres or halves, so we call them the cerebellar hemispheres. So there's a right cerebellar hemisphere and a left cerebellar hemisphere. And then the cerebrum is the cerebral hemisphere. So cerebral, cerebellar. The hemispheres are joined at the center by the vermis. The vermis is like just a little band of cortical tissue here in the middle. It's easier to see on the the ventral and inferior side of the cerebellum. It's not easy to see on the top. There are fissures, right, or little sulci, little grooves that are present, and the area between the fissures is referred to as a folium, or plural is folia. It's literally just folds. So there are little folds that are present. So the fissures are the little grooves, and then the folia are all of the raised areas or little folds that you see. And again, the reason that it's folded like that is to just increase surface area. So increase the amount of neurons that you can put out there. The gray matter is superficial. So all of this on the outside is gray matter, where we have the neuron cell bodies. That gray matter is the cerebellar cortex. And then the white matter is located centrally. So all of this white in here is the white matter. The white matter is referred to as the arbor vitae, or tree of life, because when you cut it, when you section it, it literally looks like a tree. This picture doesn't show it as well, but there's like the trunk coming in, and then there's all these little branches that branch out, and it literally looks like a tree. The arbor vitae, all of this central white matter that you see here inside the cerebellum does have nuclei present. Remember, a nucleus is just a mass of neuron cell bodies. So there are little nuclei that are surrounded by white matter inside the cerebellum. The most important of these is the dentate nucleus. The dentate nucleus is what you see in green here. Dentate, like the teeth, dental. Somebody thought that these looked like little teeth sticking off of it. So that's where the term dentate nucleus comes from. The dentate nucleus is involved in the planning, initiation, and control of voluntary movements. Which we know the cerebellum is highly involved with learned movements, right? Perfecting repetitive learned movements. Other nuclei, vestigial nuclei, interposed nuclei, there are numerous. You can kind of see them down here, the little kind of dark circles. There are numerous nuclei, but the dentate nucleus is the largest and most important. So here you can see the cerebellum. So in the first picture here, you see the brain stem, right? The pons is this kind of swollen belly of the brain stem right here. Up top, you've got the midbrain, the pons, and then underneath that's the medulla oblongata. So directly across from that, you see first the open space is the fourth ventricle, and then behind the fourth ventricle, you have your cerebellum. Again, the cerebellum is highly folded. All of the little folds are folia or a follis for just one of them, right? So you see all the little folds or folia. If you look where it's sectioned, both of these pictures where it's sectioned, you can see the gray matter on the outside, the cerebellar cortex, and then on the inside, you see the white matter, right? The arbor vitae. The dentate nucleus is this large nucleus that's present in the center of the arbor vitae. You can see in this picture here, we have the two cerebellar hemispheres or halves. In the center is the vermis, and then you can see the dentate nucleus on either side. In this picture here, again, you can see actually, you can see it better at the bottom, but you can see the vermis, or this band of cortex that goes down the center of the cerebellum. There is also what's called a primary fissure, which is this kind of more prominent fissure that you see right here, and that does separate the cerebellum into an anterior lobe and a posterior lobe. So you see the anterior lobe up here and the posterior lobe in the back. All right, so we will look at more specific regions of the cerebellum. The cerebellum has multiple developmental regions. So like as we look at evolution, there are regions that have been there longer, right? In evolution, and there are regions that are newer and then even newer. So the oldest part of the cerebellum is the floccular nodular lobe. The floccular nodular lobe is connected to the vestibular apparatus. So we refer to it as the vestibulocerebellum. So you can imagine this is involved with balance. The anterior lobe was the next to come developmentally, like in evolution. And the anterior lobe is connected to the spinal cord. We refer to this as the spinocerebellum. So if you look at the picture here, the vestibulocerebellum is in the very center right? This is the area having to do with balance that was, first evolved in us. Then again, the spinocerebellum is what you see on either side of that in green. That was next to develop. And then the posterior lobe is the most recent developmentally, and it's connected to the cerebral cortex through the pontine fibers. So we call it the pontocerebellum or neocerebellum, and that is on either side like this. The cerebellum coordinates information from the cerebrum, from the spinal cord, from the brainstem. So there are tracts or connections of white matter where axons are traveling back and forth between all of these areas and the cerebellum. So the cerebellum has a lot of axons coming in because it's getting a lot of different information, and then it has fibers that are going out to, give this information to, the cerebrum, for example. The cerebellum, the connections, like the three areas where it's connected, physically connected to the brainstem, are the cerebellar peduncles. So there's a superior cerebellar peduncle up top, a middle cerebellar peduncle in the middle, and then on the bottom, an inferior cerebellar peduncle. So these are actual like physical connections. Inside these peduncles, there's white matter, right? So there are axons, tracts that are going in and out of the cerebellum. So the peduncles are physically attaching the cerebellum to the brainstem, but there are like functional connections. There are axons or tracts that travel from the cerebellum or between the cerebellum and the pons. Like, for example, these pontine mossy fibers you see going from the pons into the cerebellum. There are also connections, though, that go to the cerebrum, right? So you can see like these arrows up here going up to the thalamus and to the red nucleus. You can see the fibers here going to the vestibular nuclei, right? So that would be how the cerebellum is involved in balance. There are also spinal tracts, right? So here you see information like proprioceptive information coming up from the spinal cord. Fibers coming from the olive nucleus in the medulla oblongata. Okay, so lots of fibers going into the cerebellum and then fibers exiting the cerebellum as well. And this is important in order for the brain to be able to learn from the cerebellum. Right, so the cerebellum's receiving information about how our motor commands, how our physical activities and movements work. And then it's gonna send that information up to, eventually, up to the cerebrum, so the cerebrum can learn from it and do, better movements the next time. The cerebellum, the main function of the cerebellum has to do with skeletal muscles and movement. So the cerebellum maintains muscle tone. It's involved in posture and gait. It's highly involved in the coordination of precise voluntary movements. So these are learned movements that require a lot of coordination of a lot of different muscles in, sequential order. So again, coordination of reflex and voluntary activity, planning of sequential movements, and correction of movement errors. Initially, what happens when we're moving is it's planned in the premotor area, and then the primary motor cortex sends the signal down, through the spinal cord to the muscles to contract. the cerebellum monitors this, right? It sees what we're supposed to do. It receives the information from the cerebral cortex. It says, hey, this is the plan. This is the planned movement. And then the cerebellum also receives information from proprioceptors, other sensory information, so that it can see what actually happened. What was the movement actually like? And then the cerebellum compares the two, right? The plan, the intended movement, and the actual movement. And the cerebellum comes up with changes that should be made to the movement, and it relays that information up to the cerebral cortex. So it's got information coming in from the spinal cord, it's got information coming in from the cerebral cortex, and it takes all of that information and integrates it, right? This is what I planned to do. This is what actually happened. These are the adjustments that should be made. And then it gives that information, right? Any mismatch between the intended movement and the actual movement is processed. And it gives that information about the error back, up to the motor cortex. The cerebellum sends it to the motor cortex so that the next time around, the movement can be better. And this happens over and over and over again. And this is why our movements get better and better and better, right? The first time you do something, it might not be very good. It's a clumsy movement. It doesn't happen just the way you want it to. But the next time you do it, you get a little bit better. And the next time you do it, you get a little bit better, right? So the cerebellum is highly involved in this perfection of learned movements. Signs of cerebellar dysfunction are movement signs, right? They're signs that have to do with muscles. So hypotonia, because there's a loss of muscle tone, right? The cerebellum is involved in maintaining muscle tone. So cerebellar dysfunction results in a loss of muscle tone. So the muscle tends to feel floppy. There's a diminished resistance to passive movement. There are typically gait changes, so changes in the way that the person walks. One, they typically walk on a wide base because they don't have coordination. You lose that muscle coordination because you don't have cerebellar guidance any longer. So movements are not coordinated. So the walking feels uncoordinated and balance feels off. So the person walks with a wide base, right, with the legs spread wide to try and, compensate for that. Also typically stiff-legged, right? So not like nice muscle movements, but stiff-locked legs to compensate for the loss of muscle tone, to try and rely on bone structure to maintain that strength as opposed to muscle tone. Also, person will lurch towards the side of the lesion when walking. I meant to mention on the last slide that the cerebellum has ipsilateral effects on muscle. So like the right side of the cerebellum is coordinating muscle movement on the right side of the body. The reason for that is as signals go from the cerebellum to cerebrum, the fibers cross. Okay, so from the left side of the cerebellum, it crosses to the right side of the cerebrum. But then remember, so we're going from the left side of the cerebellum up to the motor cortex on the right side. Remember that when the motor cortex sends signals down to the muscle, it crosses again. So it crosses back over to the same side. So right side of the cerebellum has coordination on the right side of the body. Left side of the cerebellum has coordination on the left side of the body. So lurches towards the side of the lesion. So if there's a lesion on the left side of the body isn't coordinated. It's not working correctly. So, the right side will work fine, but the left side doesn't. So the person kind of lurches or falls towards that side. Ataxia can occur. Ataxia, there's just a loss of muscle coordination. And this leads to decomposition of movements, right? So breakdown of movements, and also intention tremors. These are tremors, but the reason we call them intention tremors is because they look like an intended movement. The trajectory of a reaching movement is lost. So you lose control of what you're trying to point at or what you're trying to grab, where you're trying to place your hands. This is what we do the finger nose test for. So the person will often overshoot the nose, right? When you're trying to put the finger on the nose, they won't put it on the nose. Or if you set something in front of them and ask them to grab it, they won't be able to actually grab it. They'll overshoot the target because they don't have that coordination and control of muscles. And here you can see all of these, right? So hypotonia, again, decreased muscle tone. So typically, like with the baby, when you pull them up like this, the head won't fall backwards like that. They'll hold their head up, right? Same thing with an adult, right? They're not going to let their head just drop down. And then in this case, this is just complete lax and floppy as opposed to having some muscle tone present. Ataxia, you see the test for ataxia, hold something in front of the child or the patient and ask them to touch it. Someone with ataxia can't just reach out and touch it appropriately. They're going to overshoot or shoot, to the left or to the right. And here, this is the gait that we see, someone with ataxia, loss of coordination. You see that their posture is kind of trying to maintain balance with the arms. The legs are wide. The steps are irregular. And they say like somebody who's walking on a boat on rough seas or someone who is drunk because the movements just are not coordinated. Other signs of cerebellar dysfunction, dysdiadochokinesis is an inability to perform rapid alternating movements, which does take some coordination. So for example, like supination and pronation. So supination, pronation, that movement should be pretty easy to repeat back and forth. Weak tendon reflexes, and then also after that, the little, weak knee jerk that comes with the reflex, you'll have a pendular knee jerk that occurs. So the knee jerks, right? There's a weak response to the knee jerk, and then it swings back and forth, whereas muscle tone would normally stop that. Nystagmus. So nystagmus is when the eyes are going back and forth, right? So this occurs because of an incoordination of the eye muscles. Now, movement of the eyes involves a lot of things, right? Like functioning the visual pathway. There's a lot that's involved with this, but it also does involve the cerebellum, the vestibular nuclei, the labyrinth in the inner ear, along with visual pathways and motor pathways. So there's a lot that's involved with coordinating eye muscle movements. But loss of cerebellum results in some incoordination of the eye muscle movements and then nystagmus. Dysarthria can occur. Dysarthria, speech that is slurred. So dysarthria is like dysfunctional speech. But the speech is slurred and jerky. Syllables are often separated from one another instead of flowing together like one word. And this is because of faulty coordination of the impulses that are going to the lips, the tongue, the palate, right? All of these things that are involved in articulation, right? So like dysfunction of articulation because of the cerebellar dysfunction. There's no paralysis, right? So no paralysis. No loss of sensation. Muscle contraction can be weak. There is like a loss of tone, but the muscles still contract, right? There's no paralysis. It's just that there's no coordination with muscle contraction. So the muscles, they don't, they're not paralyzed, so they don't completely atrophy and wear away. They're still there. They're just not coordinated. And there is some loss in tone, so like some weakness in that sense. But loss of coordination is just huge. Okay, we'll go ahead and stop there since we're completely changing topics here, moving on to the thalamus and then the brainstem. So we'll stop here and then we'll do the rest of the lecture next time we meet.